Sundgreen. Discussion of ILB, novel therapeutic agent for ALS. Dr. Sundgren, please proceed. Thank you.
Thank you very much, Dr. Brown. It's let me see, is this in full screen presentation now or?
Not now, it was a minute ago.
Okay.
Great.
Thank you very much. Thank you for the organizers for letting me presenting this promising new therapeutic candidate for ALS. My name is Claus Sundgreen. I'm the Chief Medical Officer of Tikomed in Sweden. It's a privately owned company located on the west coast of Sweden. So please just allow me a second with some technical issues. Boom, boom, boom. Here we go. So in medical treatment, dextrons are used for volume for blood clots. Sorry, I have some issues here. I can't see my screen. Just sorry, Dr. Brown. I just need another second to get the screens up and running. I can't see my own screen.
Thank you, Tom.
Oh, what's going on here?
If you're able to see your screen in the non-presentation mode.
Yeah, probably we'll do it this way. That's probably easier. Sorry for that.
No problem.
Let's go to the slide number 2 here. So in medical treatment, dextran are often used to prevent blood clots and act as a remedy to increase blood volume, especially after significant fluid loss or bleeding. However, ILB is a new direction in this field. It's specially designed. It's a highly sulfated, low molecular weight version of dextran, modified to potentially aid in treating nerve-related and muscle-related conditions. It's important to understand that ILB is still in the experimental phase and hasn't yet been approved for treating any diseases. We are exploring its potential benefits, but its safety and effectiveness are still under investigation and haven't been officially confirmed by regulatory authorities. For our clinical trial, ILB will be supplied as pre-filled syringes for once weekly dosing. ILB is a platform. It's showing promise as a versatile treatment across several diseases. And the safety and the tolerability of ILB in ALS have been investigated in phase 2A clinical trials in ALS and are now ready for advancement to the next phases of clinical trials. We have moved forward into phase 2 in islet transplantation, a potential breakthrough in treatment of type 1 diabetes. We have obtained promising results in preclinical models of stroke, traumatic brain injury, glaucoma, and cardiovascular diseases. So amyotrophic lateral sclerosis is a serious condition that leads to progressive muscle weakness due to the loss of motor neurons in the brain and spinal cord. Although we've learned a lot about ALS, we still don't fully understand what causes it. Researchers have identified several factors that might contribute to ALS, like abnormal protein folding, mitochondrial problems, excessive glutamate causing harm to neurons, inflammations in the nervous system, and oxidative stress. Despite these insights, we still don't know why some people get ALS. Treatment so far has targeted only some of these issues, which means they have had limited success in changing the course of the disease. This highlights the need for treatments that can address the many different factors involved in the pathogenesis of ALS. ILB directly or indirectly targets more of the disease processes involved in ALS by modulating the release of hepatocyte growth factor. Hepatocyte growth factor, or HGF, was first identified in the liver, is a growth factor that is involved in numerous processes, including the development and maturations of the nervous system during growth. In adults, it acts as a repair signal, helping cells recover from injury and preventing them from dying prematurely. It acts like a repair kit, keeping the cells healthy and function properly. HGF acts via the MET receptor complex on the surface of the cell, and the downstream effect reduces oxidative stress, mitochondrial function, programmed cell death, toxicity caused by excessive level of glutamate, and in addition, HGF reduces inflammations in the glial cells, the cells supporting the nerve cells in the brain and spinal cord. In animal models, we have seen that activations of the HGF receptor delays onset of the disease, prolongs the lifespans, and delays the loss of motor neurons and improve motor function. So the mode of action of ILB has been investigated in cell and animal models. Heparin binding factors, including HGF, are stored in extracellular matrix of tissues, and ILB competes with this binding of these factors. Upon dosing of ILB, heparin binding factors, including HGF, are modulated and released, which initiates the production, release, and activation of a cascade of pro-neural growth factors, resulting in restorations of mitochondrial function, neutralizing of the toxic effects of excess glutamates, and resolves inflammation. Turning to the clinical experience with ILB, then ILB has been tested in human volunteers in doses up to 24 milligrams per kilo body weight before clinical trials in ALS were started. 2 phase 2A trials have been completed in ALS patients. Both trials were open-label, single-center trials with the objective to determine the safety and tolerability of weekly subcutaneous dosing of ILB in patients with ALS. Secondary endpoints were exploration of efficacy in terms of functional status, quality of life, and the exploration of various biomarkers. The TM105 study, also known as the Sundgreen study, was undertaken from October 2018 to March 2020 and explored a dose of 1 milligram per kilogram of ILB given a subcutaneous injection every week. The study was sponsored by the company, by Tikomed, and the second two trials, the RD7/250, the 250, or the Birmingham trial, was an investigator-initiated trial sponsored by the University of Birmingham in the UK. This trial explored 2 milligrams per kilogram of ILB, also dosed weekly. In this trial, patients were initially screened for 10 weeks, with extension plans of additional 14 and 24 weeks to a total of 48 weeks. The inclusion into this trial was terminated prematurely, unfortunately, due to the COVID pandemic. The main eligibility criteria for the 105 study were clinical diagnosis of ALS and forced vital capacity of at least 65% or more of the predicted for age, height, and gender. For the Birmingham trial, the entry criteria were diagnosis of definite ALS by the ALS score criteria and a forced vital capacity of 50% or more. There were no restrictions in terms of time since first symptoms or requirement of specific value of ALS FRS score at the entry point. For both trials, pregnancy, severe concurrent diseases, chronic infections, or addictions were exclusion criteria. Due to the known anticoagulant properties of dextrans in general, known bleeding orders, recent major bleeding, or treatment with anticoagulants was not allowed in the trial. When these trials were started, there was a theoretic concern of an interaction between ILB and riluzole, and thus riluzole treatment was not allowed in these two clinical trials. 13 patients were enrolled in the 105 study, and 11 out of 15 planned were enrolled in the 250 trial. The median age was 58 and 57 years, respectively, which spanned from the young to the elderly. There was an overweight of males in both trials, 3/4 in the 105 trials and 2/3 in the 250 trial. Time from diagnosis to trial entry is best illustrated by the median of 6.7 and 4.7 months, as there is one trial participant in the 105 study that was diagnosed 6.5 years before the first visit. In the 250 trial, one patient had a family history of motor neuron disease, while there's no information from the 105 trials of family disposition. Genotyping was not performed in any of the trials. Overall participation in both trials, participants in both trials had preserved lung function with a median of 85 and 75% of predicted, and a functional status preserved to a certain extent with baseline ALS FRS-R of 36 and 38.5. In the 105 study, one patient reported prior use of riluzole, while 8 patients in the 250 trial had a history of riluzole use. One of those had discontinued riluzole before starting the trial, while 7 patients had their riluzole treatment discontinued in order to enter the trial. The 250 trial was originally planned as a 10-week trial with a 14-week extension that participants could enroll in if not meeting the protocol specified stopping rule. An additional 24 weeks extension of dosing was added to the study, as well as a long-term follow-up, which took place 21 months after study start. Of the 11 patients, 9 participated for 10 weeks. 1 chose not to enroll in the 14 weeks extension. 6 patients reached the 24 weeks time point, which here represented with the second line of green marks, and opted in for the next 24 weeks of the trial. However, in March 2020, the study was halted due to COVID pandemic. In this trial, this first trial, the patient visited the site every week for injections of the medication. And as the hospital was closed due to the pandemic, the trial ended. Remote follow-up was performed up to 76 months after enrollment in the trial. The primary endpoint of both trials were safety and tolerability. In the 105 trial, a low concentrations of 20 mg per ml was used, and in the next trial, a 5 times more concentrated solution of ILB was used. In the 105 trial, a total of 24 adverse events were reported by 11 of the 13 patients included in the 5 weeks treatment period and the 9 weeks follow-up period. The number of AEs judged to be related to ILB was 7, and no serious adverse events were reported in this trial. In the 250 trial, all patients reported at least one adverse event, and a total of 270 adverse events were reported. Of those, 98 were just to be related to the drugs, and these were mainly bruising at the injection sites. One serious adverse event was reported, muscle weakness, which was not related to ILB and resolved without sequelae. In the long-term follow-up period, 3 patients passed away, all more than 52 weeks after the last dose of ILB. Looking at the detailed report of adverse events, the most common report in the 105 trials were falls and nasopharyngitis. In the 250 trial, the most often reported adverse events were laboratory tests out of range, reported in 95 instances in 10 patients, followed by bruising at the injection sites, reported by 9 of 11 patients and in 92 instances. High cholesterol, falls, and infections were reported by half of the patients. And as previously mentioned, bruising was just to be related to the study drug. All reported adverse events were mild or moderate of intensity. Of note for the next slide, there were 2 reports of bleeding-related events. 1 event of bleeding at the injection site and 1 event of skin bruising not related to the injection sites. And these were observed in the 105 trial. So, and another important safety aspect in the trial was the risk of bleeding. As mentioned previously, dextran with larger molecular weights has an anticoagulant effect and a pronounced effect on the activated partial thromboplastin time, a measure of how long time it takes blood to clot. In both trials, APTT was measured serially in the hours after the injections of ILB. The data presented in this slide is from the 105 trials, and no difference was observed between the 2 trials. At 2.5 hours after the injection of ILB, the clotting time is prolonged by 7 seconds, from 26 seconds to 33 seconds, which is still within the normal range. Before the start of the ALS trial, ILB had only been tested in human volunteers, and to investigate the relationship between administration of ILB and the release of hepatocyte growth factor, serial measurements were performed after injection of ILB, answering the question of does ILB release HDF when dosed to ALS patients. The left side of the y-axis is the concentration of ILB, and the right side is the concentration of HDF in blood. The blue lines are data from the 105 study using a dose of 1 mg per kilogram, and the red lines are data from the 250 trial using a 2 mg per kilogram dose. The solid line represents the concentrations of ILB, and the dotted line the concentration of HDF released to the blood by ILB. By inspection of the curves, it's noted that there is a nice correlation between the concentration of ILB and the release of hepatocyte growth factor, and also a dose response is observed. Please note that these data are obtained from 2 different trials in 2 different populations, resulting in variability of the data. So coming to the effect sizes, to the clinical effect in the 105 trial, ALS FRSR was the primary efficacy endpoint. Patients were treated with weekly doses of ILB for 6 weeks and followed for up to 99 days. ALS FRSR were obtained weekly at the dosing visits, with less frequent visits in the follow-up period. The graph displayed the change in the mean value of ALS FRSR in these patients. After the first dose at day 1, the ALS FRSR increases over the next 3 dosing visits, stabilized at the last dosing visits at day 29, and wear off over the next 3 weeks until the last dose, after the last dose and until the last visit at day 99. Looking at the individual trajectories of ALS-FRSR. 11 of the 13 patients has scores about 32 at the start of the trial, while 2 patients have markedly lower scores. In these 2 patients, a consistent increase in ALS-FRSR are noted from start to visit 5. We have analyzed the individual domain and items of the ALS-FRSR without identifying any clear pattern of changes in these values of the individual items. And the next trial in the 250 trials, the ALS scores displays in this slide. The solid line represents patients that receive all weekly treatment as per protocol, and the dotted line patient that had 1 or more missed doses. The maximum number of doses that any patient received was 38 over a 48 weeks period. The maximum number of doses, the blue line is the trend line with the gray arrow representing the gray area representing 1 standard error. In this trial of longer duration, the scores of ALS-FRSR appears to be stable with no clinical significant change. However, 2 patients, patient number 2 declined a 7-point experienced a decline of 7 points over 46 weeks, equivalent to a decline of 0.7 points per month, and patient number 6 experienced a decline of 6 points over 24 weeks, equivalent to approximately 1 point per month. A number of exploratory biomarkers were measured in both trials. In the 105 trial, no difference was observed in the level of neurofilament light change from the start of the trial to the end of the trial over 5 weeks. The NFL values from the 250 trials are displayed in this slide on the left-hand side. Also, the urinary P75 value was measured in this trial, but these are not presented in this presentation. In general, the data suggests that there was no change in the disease biomarker in treated patients during the 38-week trials. However, the sample collections was variable, primarily due to differences in duration of treatment and follow-up for each patient. On the left-hand side, on the right-hand side, the value of N-acetyl aspartate is presented. So N-acetyl aspartate is synthesized exclusively by neuronal mitochondria, and the raised level observed in ALS patients are believed to reflect neuronal damage and mitochondrial dysfunctions. Both in the 105 trials and as here displayed here in the 250 trial, treatment with ILB reduced the serum level of NNA, which we believe reflects improvement of mitochondrial function in this ALS population. In conclusion, the pleiotropic effect and multifaceted effect of ILB addresses several steps in the pathogenesis of ALS, reducing neuroinflammation, mitochondrial dysfunctions, and glutamate toxicity. Safety and tolerability of ILB has been established in these 2 presented phase 2A trials. In the evaluation of the clinical effect demonstrated, it should be acknowledged that both trials were open label with associated limitations of the interpretation of the results. However, we find the results encouraging, and the next step is to take ILB to phase 2B trial, which will be planned in collaborations with our newly established scientific advisory board. Please let me extend my gratitude to the patients, the caregivers, the family affected by the dreadful disease for the participating in the clinical trials, and also let me thank the teams at Sahlgrenska, Birmingham, and at Tikomed that made these 2 trials happen. Thank you very much, and apologies for the technical difficulties.
Not at all. Thank you. Very, very nice backward review of these studies. We look forward to learning more. Let me suggest that we take just a very brief break and reconvene at 3:05 for the panel. I look forward to further discussing these as an ensemble of really incredibly interesting presentations. That's all right with you, Jen? That's perhaps how we should proceed.
It sounds wonderful.
3:05. Good.
That's
Thank
fantastic. Yep.